Hi, this is Rich, originally from Cincinnati, Ohio, and welcome to this week's discussions on Genomics and Informatics. What's around the corner? With an innocuous appearing two-page article in the 1953 issue of Nature, American James Watson and Brit Francis Crick described the structure of DNA for the first time. This double-stranded molecule, composed of 4 base pairs and located in the nucleus of the cell, contains instructions governing the development and functioning of all life. DNA is organized into long structures called chromosomes. Chromosomes are further divided into genes, the basic unit of heredity. DNA technology has proven itself to be useful in interesting ways in the past few decades. In addition to solving crimes and freeing falsely accused prisoners, the application of DNA research has allowed us to solve some of history's greatest mysteries. United States Air Force First Lieutenant Michael Blassie was killed in action in Vietnam on May 11, 1972. Government officials were certain enough of his anonymity to inter his remains in 1984 at Arlington Cemetery in the tomb of the unknown soldier. Using a combination of historical research and mitochondrial DNA, Blassie's remains were identified in 1998 and returned to his family. Similarly, scientists have been able to combine history and DNA analysis to support theories that Thomas Jefferson fathered a child with a slave he owned named Sally Heming. In 1918, Bolsheviks in Russia assassinated the last Tsar and his royal family. There were rumors that the eldest daughter, named Anastasia, survived the assassination and escaped. These rumors spawned everything from movies starring Ingrid Bergman and cartoons to claims to the Russian throne and its treasures by a woman claiming to be Anastasia. The most notable of these petitions was made by immigrant and American citizen Anna Anderson, who died in 1984. Using a piece of preserved intestine from a procedure, scientists proved after her death that Anna Anderson was not Anastasia. Subsequent DNA research accounted for all of the Romanovs, who were buried in mass graves near the site of their deaths. Since the discovery of King Tut's tomb in 1922, questions have existed about his lineage. Recent DNA analysis of royal mummies have confirmed that King Tut's father was the pharaoh Akhenaten, and his mother is a woman simply known as the Young Lady, who is Akhenaten's half-sister. Additionally, researchers have conducted a genetic autopsy of Tut. Combined with other forensic data, it is suggested that he died from complications associated with malaria. He suffered from the genetic disorder Kolar disease 2, as well as scoliosis, pes planus, and mild clubfoot. Recent data suggests that King Tut was not the spear fighter you see here on the left, but a fairly compromised invalid who found it difficult to walk and spent a lot of time sitting, as you see here on the right. While all of these DNA discoveries are interesting, we are concerned with preparing ourselves for the future of genomics informatics, the place at the intersection of clinical practice, technology, and genetics. Within this course is our golden opportunity to try to anticipate some of the things we will need to know in order to take care of our patients in the future. In 1990, Dr. Watson, one of the aforementioned discoverers of DNA, who you see here, managed the beginning of the Human Genome Project. This project's goals were to identify all of the 25,000 human genes, determine the sequence of the 3 billion base pairs that make up human DNA, store the information in accessible databases, improve tools for analysis, make the data accessible to industry and the public, and address the ethical and legal issues associated with this information. This international effort was completed in 2003 at a cost of $10 billion, using the DNA of about 10 people. The International HapMap project is the next big step in genomics. Humans share 99.9% of their DNA with their next door neighbor. Identifying that 0.1% that makes us different from everyone else is the next big step in DNA research. Instead of DNA sequencing just a few humans' genomes, the HapMap project will sequence carefully selected individuals from all over the world so that we can understand what the variation is between humans. Our understanding of DNA sequencing has exploded. In the future, DNA sequencing that took 13 years is only expected to take a few hours or less. Dr. Francis Collins, who you see in the upper right-hand corner, succeeded Watson as the director of the Human Genome Project and is the current director of the National Institute of Health. He believes that it will only be 5 years before an individual can have their DNA sequenced for $1,000. An individual's entire genome would fit on a CDR or a 1 gig thumb drive.